Okay. So yes, how many of you remember those phases? Raise your hand. Yes, how many of you remember? Phases of meiosis. 4 phases of meiosis. Who can tell me the name of those phases? Just name. Who can tell me? Not like this, not like this. One student at a time so that I can hear it clearly. So yes, who can tell me? Just raise your hand. Or you can stand up. Those who want to tell, just stand up and speak up. Yes. अभी सारे इकट्ठे तो बोल रहे थे हैं जब एक का बोला तो सब चुप हो गए व्हाई सो okay so last bench middle row girl in the middle beta stand up and tell me middle row last bench middle girl yes beta so can you tell me the 4 phases of meiosis that's good sit down beta that's good now who can tell me the 5 phases of prophase 1 now prophase 1 के 5 phases जो हमने पढ़े थे who is going to tell just the name just raise your hand okay I will call don't worry about this so I would like to call corner row last bench boy in the corner towards wall yes beta. Tell me the names only the names of prophase 1 phase ठीक है 5 phase के नाम बताओ with sequence in sequence yes beta pick up एक मिनट बेटा बाकी सब प्लीज बी क्वाइट लेट मी हियर प्लीज be quiet everyone there is lot of noise that is coming please be quiet pin drop silent yes that's good so yes beta now speak okay do one thing beta थोड़ा सा आगे आ जाओ come forward can please come forward yes now stand here beta यहीं खड़े हो जाओ yes so yes lactoline but lactoline comes before or after of zygotine do you remember that short trick that I have shared with you तो लास्ट सेशन में मैंने आप सबके साथ एक निमोनिक शेयर किया था इसको किसको याद है Who remember that? Yes, first bench corner girl. Yes, beta, you are raising your hand. Yes, first bench. Yes, beta. Yes. So, on the basis of that, can you tell me the name of those phases?
Leptotene, zygotine, pectine, diplodene, and dialysis.
That's nice. That's nice. So now, beta, जो अभी आप खड़े हो, आपको पहले बताओ 5 phases. Do you remember those phases name? Do you remember? Lata means lactonine. Zara, zygotine. Pani, pachetine. Day, that is diplodene. And Dina, that is dikinesis. आपने नोट किया था बेटा? Okay. So go and sit. Please उसको एक बार फिर से पढ़ो because we are going to take some questions on that one. ठीक है everyone? I'm giving you 2 minutes to go through these. ठीक है? और उसके बाद take out your worksheet. Then we will solve the worksheet. ठीक है? पहले कुछ फटाफट phases को पढ़ो. What were those phases? What we discussed in the last session? But please be silent. Be silent without making any noise. Just go through that. So I hope this much time is sufficient, है ना? This much time is sufficient. So let's proceed, beta. ठीक है? So we are proceeding, and now you all are on unmute. What you have to do, you have to maintain the discipline. Please do not speak up unless I call your name. ठीक है? So we can understand this thing because this topic is very important. ठीक है? I want you to make it, easy. ठीक है? इसको easy बनाओ. कैसे बनाओगे? Please pay attention. Okay? So now let's move ahead. So we learned about meiosis and mitosis. I think you already did in your class. Class में हो चुका है आपके mitosis. So in mitosis, equal number of chromosomes, okay, or cells form. So now, mitosis usually happens in which type of cells? Who can tell me? In which types of cell usually mitosis take place?
Somatic cells.
Somatic, haploid or diploid? Somatic, haploid or diploid? Diploid. Yes, you're right. It's somatic diploid cells. So it is the always case. क्या ये हमेशा ही ऐसा होता है? और is there any exception? क्या सिर्फ माइटोसिस सोमैटिक डिप्लॉइड सेल्स में ही होता है और इज देयर एनी एक्सेप्शन टू दिस? Yes, is there any exception? Raise your hand. Those who are saying yes. Is there any exception, beta? I'm just asking, is it the correct or not? Just raise your hand. If you are saying yes, there is an exception. They are saying that it means you didn't raise your hand. It means you are saying it only happens in somatic diploid cell. But who is yes? किसी ने कुछ बोला है yes बेटा कम जल्दी आओ. अब मुझे देखो ऐसा है कि टीचर को भी तो आपने कुछ बताना होता है. It's not teacher's duty to make you learn something. Yes, yes, speak बेटा. First bench girl. I think she is. Yes, sir.
It happens in germ cells during cell division.
In plant cells during cell division. Okay. Little bit right. Okay. Or any other exception, beta? In drawn cells. Okay. In drawns. Okay. That's also nice. So that's good. That's good. So let me tell you, drawns. Drawns are what? Everyone note down. It's in your NCRT, beta. It's in your NCRT. It's in your textbook. It is given this line. In honeybees, drones. Drones are what? Male bee. Okay? Drones are male bee. They are haploid. They are not diploid. Okay? They are haploid means they have only N. Okay? While here queen bee. Queen bee, female bee is diploid. And it gives birth to two types, or we can say produce two types of offspring. One is drone, okay, that is haploid, and second is worker bee. worker bee, that is also female. That is also female and it is diploid. So here you can see we have two females, both are diploid, but male. Only the male is haploid here. In this haploid drone, cells divide through mitosis. Here, cells divide through mitosis. So here we can see the exception that mitosis can take place in haploids also. ठीक है, ये haploids में भी हो सकता है. It means that. ठीक है. And drone, queen give birth to drone also. This drone produced by queen bee through unfertilized eggs. Unfertilized eggs. Okay. Through a process that you are going to learn later. And आपने 9th class में भी थोड़ा पढ़ा होगा. That is parthenocarpy. Here, eggs are unfertilized in case of drone from where it is raised. It is developed from unfertilized egg through a process called parthenocarpy. Okay? Is it clear, beta? जो आपके एनसीईआरटी के टेक्स्ट बुक में एक लाइन दे रखी है, this is how we can explain it. That honey bee जो है एक मेल एक फीमेल. Male is called drone, female is called queen bee, and it produces two types of egg. ठीक है, unfertilized, and from one it gives birth to the worker bees that are also female and they are diploid. But male bee is haploid. Okay. So note down this. You will not get this anywhere. Okay. So note down this. And next, in plants also. In haploid cells. Okay, mitosis can take place. Not in all plants. Okay, not in all plants. Can you tell me the types of plants? Okay, those who can flower. Flowering plants को क्या कहते हैं? You tell me the name of flowers. Sorry, plant type. We have pteridophyte. We have gymnosperm. So flowering plants को क्या कहते हैं? What do we call it? Angiosperm. So in angiosperms, in their haploid cells, ठीक है, this mitosis can take place. ठीक है, but when it depends upon their cycle, is it sporophytic or gametophytic, that you will learn during plant kingdom. ठीक है, in detail you are going to learn about this in plant kingdom. For right now, just remember in plants angiosperm, क्योंकि उसमें क्या होता है, ऑल्टरनेशन ऑफ जनरेशन. सबने ये पढ़ा होगा पहले, क्योंकि आपकी एनसीईआरटी की बुक में इट इज गिवन एज यू लर्न इन योर प्रीवियस क्लास. ठीक है, तो इट इज you all are already aware of this thing. So alteration of generation means haploid to diploid or diploid to haploid, kind of this. ठीक है, so इसको हम क्या कहेंगे, जो haploid stage है, in that cells divide through mitosis in that case. So in plants also, ठीक है, so in plants also haploid undergoes mitosis. ठीक है, have you noted down this thing? Yes, beta?
Yes.
Okay, that's good. That's nice. ठीक है, so this is what I want you to know. ठीक है, so now let's go back to one more thing, that in plants, can you tell me pollen grain is diploid or haploid? Pollens are. Any of you can tell me pollens are haploid or diploid? They are haploid. And then how do they grow or they divide? Yes, any of you?
And mitosis.
Mitosis, because we are learning about mitosis, so it's easy to say, huh? It is easy to say because we are learning about mitosis. So yes, pollen grains also they produce two cells, vegetative and generative cells, and then generative cells also give rise to two male gametes. Okay? So that we will learn when we will discuss in detail plant kingdom in your upcoming classes. Okay? So now take out your worksheet. So first question, everyone, pay attention to the first question. So in this cell cycle is given, beta. Cell cycle is given here like this. Okay? And it is mitosis that is happening here. It's the mitosis. Okay? Now everyone pay attention to this question. ध्यान से इस क्वेश्चन को देखो. Everyone pay attention to this question now. Everyone pay attention to this question. So given below is a schematic breakup of the phases or stages of cell cycle. Which one of the following is correct indication of the phase or stage in the cell cycle? So it is given here. Okay. So B. They are saying B is metaphase. They are saying B is a metaphase. Is it the metaphase? B is a metaphase. So B. There are 4 stages in the mitosis. Are you aware of this now? There are 4 stages in mitosis as we discussed at the beginning of the session. जो meiosis की थी वही stages mitosis की है ना? So here on at third number which phase come? कौन सा phase आता है third number पे in mitosis? Is it the metaphase that comes at third number? So is it right? First statement is right or wrong? So it is anaphase. It is not metaphase. Okay, so it's wrong. Coming to B. C is karyokinesis, nucleus breakdown, the splitting of nucleus. Is it correct? No. Why it's not no? Because yes, beta. Because it is not here. Okay, at C. C is what? C is what? So it is C is cytokinesis. Here cytokinesis happening. Coming to C, its D is synthetic phase. D is synthetic phase? Yes. Yes. So it's G1 phase, it's S phase, and this is G2. So yes, it's synthetic phase. So it's correct statement. So it's correct. Mark it. Everyone mark C is the correct answer. Now you don't have to do second number question at present. Okay. So in second number question, we will do. Achha, there is actually printing mistake. Okay. So just come to this A part. Okay. So this diagram is given from the event of mitosis. Okay. माइटोसिस के जो इवेंट दिया गया है ठीक है यहाँ पर and you have to tell which statement out of A, B, C, D is correct. So what is shown here, beta? It's a cell. Okay, cell is given. This is the arrangement. ये वाली शेप हमें कब देखने को मिलती है? When we observe this shape. ओके, those who know the answer, बेटा just खड़े होके आंसर दे दो. ऐसे आपस में इकट्ठे होके बोलोगे I am not able to understand this and you are also wasting your energy. It's easy. Just stand up and tell. ये आप सब खड़े होके मुझे आंसर दे रहे हैं. ये मुझे खड़े होके आंसर मिल रहा है. या मैंने गलत बोला या आपने गलत किया. Who did this? हाँ, इसको आंसर आता है. Those who know the answer, make it clear to everyone, ताकि हम उसको डिस्कस कर सकें. आई नो यू नो द आंसर, दैट्स रियली गुड बेटा, बट आई वांट कि कोई एक उस चीज को रिप्रेजेंट करे. ठीक है, जिसको आंसर आता है जस्ट स्टैंड अप एंड टेल मी द आंसर. ठीक है, नाउ आई वांट फ्रॉम द कॉर्नर रो. ओके, थर्ड लास्ट बेंच, मिडल बॉय, आ जाओ बेटा. थर्ड लास्ट बेंच, यस, थर्ड लास्ट बेंच, मिडल, यस बॉय. तो बेटा, what is this? Which stage is this? अभी इसको options को छोड़ो, leave the options. Just tell me which stage is this? You can see chromosomes are aligned here. Chromosomes are aligned here. Which stage is this? Yes, beta? Metaphase. So what is happening here? This is metaphase. You are right. Why it is metaphase? Why we are not calling it anaphase? Can you tell me what is the difference? Why are you calling it metaphase, not anaphase? Getting answer from your partners or neighbor doesn't make you understand. है ना? I can see each and every student what they are doing. I have big screen. So see here, the first thing, align. Where they are aligning? Equator. अर्थ का जैसे इक्वेटर होता है, अर्थ का भी इक्वेटर होता है ना, मिडल इक्वेटर, एंड दीज आर पोल्स. अर्थ का ये इक्वेटर है. सिमिलरली, okay, cell has equator. This is the equator where these chromosomes are aligned, equatorial plate. ठीक है, so this arrangement makes us clear that it belongs to metaphase. And that's why answer is C. Chromosomes move to spindle equator. Chromosome made up of two sister chromatids. So these are the sister chromatids. Okay, sit down, beta. Sit down. Now coming to the third question. Okay, coming to the third question. During the mitosis, endoplasmic reticulum, ER means endoplasmic reticulum, nucleolus and nucleolus begin to disappear at. So when they begin to disappear, yes, when endoplasmic reticulum nucleolus begin to disappear, सब ध्यान से तो देखो, look here. I'm also making you recall the concept. ठीक है, everyone pay attention here. I'm just adjusting the brightness. Is it clear to everyone now? Yes. Okay. Now it's more clear, I think. Okay. So now everyone look here. We know about the interface. है ना? We know about the interface. Your cell is not dividing. ओके, जहाँ पर सेल डिवाइड नहीं कर रहा, और वहां पर जो हमारा जीनोम होता है, जेनेटिक मटेरियल होता है, किस फॉर्म में होता है? इट इज प्रेजेंट इन विच फॉर्म? यस, यस, इन विच फॉर्म इट इज प्रेजेंट? क्रोमोसोम, क्रोमैटिन और क्रोमैटिन. यस, फर्स्ट. ओके, फर्स्ट बेंच गर्ल बताइए. चलो, मिडल रो फर्स्ट बेंच गर्ल हु इज राइटिंग समथिंग. No, not you. Another side, corner girl. Yes, beta. You did. So in interface, which type of, or our genome present in which form? Chromosome. Is it clear to you? This image is clear to you, beta? Yes, beta. what is written here? What is written here? Yes, beta. Can you read it? What is written here? What is this word? What is this word? Chromatin. Chromatin. So our genome is present in the form of chromatin. Mess up. सब खुला हुआ. Last time मैंने आपको बताया था. Not in organized form. ठीक है? सब ऐसे जैसे ऊन का गुच्छा पड़ा हुआ है. Just like that. ठीक है? So that is chromatin. And you can see there is nucleus also. आपको क्लियरली न्यूक्लियस की वॉल दिख रही है, पोर्स दिख रही है. यू कैन सी इन द न्यूक्लियोलस हेयर, एवरीथिंग इज असेंबल्ड हेयर. कुछ बाकी जो चीज है वो इधर उधर नहीं दिख रही, सब ऑर्गेनाइज है बाकी सब. ओके, इट इज इंटरफेस. ठीक है, दिस इज इंटरफेस. नाउ द क्वेश्चन इज इन विच फेज दिस न्यूक्लियर एनवलप, न्यूक्लियोलस, एंडोप्लाज्मिक रेटिकुलम डिसअपियर. तो इसका मतलब कौन सा फेज होगा? In which phase it get disappear? अभी दो फेज दे रखे हैं बेटा, you have to tell me. ठीक है, I am showing you the result a kind of thing. ठीक है, this is prophase, this is metaphase, and this is the events that is happening in prophase and metaphase. Can you look here carefully and tell me in which phase you can see there is no nucleus, there is no nucleolus and endoplasmic reticulum? कौन से फेज में नहीं है बेटा? प्रोफेज में या मेटाफेज में? प्रोफेज में नहीं है. और यू कैन सी कुछ जगह नहीं है. इट इज गेटिंग क्या? देयर इज डिफरेंट. राइट नाउ इट इज प्रेजेंट बट नॉट एज मच एज इट वाज इन इंटरफेज. जो भी हमने इंटरफेज देखा उसमें पूरा प्रेजेंट था. नाउ हियर यू नोटिस, if you see here, so here it is now getting disintegrated, खत्म हो रहा है. बीच में ये देखो पोर्स बहुत ज्यादा गैप आ गया है. Earlier it was like this, it was like this. तो इसका मतलब कौन से फेज में ये जो खत्म होना शुरू होता है? It is phase. It is shown here also, बेटा. It is shown here also. You can see here from here also. So here chromosome starts to condense. मतलब जो हमारा क्रोमेटिन जो गुच्छा था वो एक बंडल बनना शुरू किया है. उसकी एक शेप आनी शुरू हुई है. कौन सी शेप है? That is X shape. That is chromosome. ठीक है. So this thing happens in prophase. अब देखो मेटाफेज में तो कोई न्यूक्लियस ही नहीं नजर आता. There is no nuclear membrane. Can you see any membrane here? So there is no membrane in the metaphase. ठीक है. So answer is it's D, early prophase. Everyone mark, it's D. Third का D answer. Sit down beta. Sit down. Now coming to question number 4. Which stage of cell division the following figures A and B represent? Yes. Which phase is this? So one stage is here. If you can figure out, एक जो इनमें से दो images दिए उनमें से एक इधर है. Now you have to find out is it prophase or it's metaphase. Which phase? Who is saying it's metaphase? Who is saying metaphase? Is it metaphase? Is it prophase, beta? It's a prophase. तो एक जो है B is prophase. It's like this. Second is what? Now it is not like this. So कौन सी phase है? Who can tell me the name of this phase? The A phase. A में क्या दिखाया जा रहा है? कौन सा phase है? Now A, अब जो A है ना ही वो prophase है ना ही वो metaphase है. It can be क्या? Anaphase और telophase. यही दो बचे. Now you have to figure out. Now it belongs to anaphase or telophase. थोड़ा सा सोचो. Think upon this. थोड़ा सा सोचो. अभी ये ऐसे है, नेक्स्ट क्या हो जाएगा? Can you see here? You see? It belongs to which phase? इसको थोड़ा सीधा कर देते. Now it belongs to which phase? Image A belongs to which phase, beta? Now sister chromatids are like this. Sister chromatids are aligned towards pole. पहले कहाँ थी metaphase में? Equator. Now in anaphase they will separated and they will move towards different poles. एक अगर ऊपर जाएगा तो एक नीचे को जाएगा. पहले कैसे थे? बीच में थे. मेटाफेज में यहाँ थे in equator. Then they split and one sister chromatid moves to this side, another move to this side, means different poles. तो ये कौन सा हुआ आंसर? Who can tell me? Now I have explained it. Now from corner row जहाँ पर girls बैठी हैं only, no boy, I can see. Okay, so here the third bench girl, I think she is sitting alone. Yes. Yes, beta. Now I have told you image A क्या है और B क्या है. Can you tell me the answer? Which one is correct? C option. Yes, everyone marked C is the correct option. I think now the fifth question will be easy. Now you are aware of all the phases कि क्या events हो रहे हैं. So now tell me, a stage in cell division is shown in the figure. Select the answer which gives correct identification of the stage with its characteristics mentioned. So कौन सा है ये? So which phase is left now? Which phase is left? We studied about prophase, metaphase, anaphase. Now which phase is left, beta? So what could be the answer of this question? क्योंकि अब अगर आप ध्यान से देखोगे, here you can see chromosomes are not clearly differentiated here. ठीक है, वो फिर से एक जो है जो कंडेंस से डीकंडेंस फॉर्म में कन्वर्ट हो गया. न्यूक्लियस इज अगेन फॉर्म हियर. फिर से न्यूक्लियस आ गए हैं, दो न्यूक्लियस आ गए हैं. ठीक है, तो इट हैपेंस वेयर इन टेलोफेज. क्योंकि एनाफेज में यू कैन सी देयर इज नो न्यूक्लियस. कोई न्यूक्लियस नहीं दिख रहा अभी आपको. यू कैन सी देयर इज नो न्यूक्लियर मेम्ब्रेन. बट एज यू अप्रोचेस टुवर्ड टेलोफेज यू कैन सी दिस स्ट्रक्चर. सो आंसर इज A option, telophase, nuclear envelopes reforms, Golgi complex reforms. ठीक है, so A. Now coming to the sixth question. अभी आ जाता है events पे that we have done. ठीक है, so question is recombination between homologous chromosome is completed by the end of. तो इसको भी थोड़ा सा रिवाइज कर लेते हैं. लेट्स रिवाइज इट. डोंट वरी अबाउट दिस. ओके, वी आर गोइंग टू रिवाइज इट. तो बेटा प्रोफेज वन जैसे माइटोसिस में होता है, इट इज जस्ट लाइक दिस. क्रोमोसोम्स फॉर्म्ड. ठीक है, कंडेंस क्रोमेटिन कंडेंस होके यहाँ पर क्रोमोसोम बना लिया है. न्यूक्लियर मेम्ब्रेन इज डिसइंटीग्रेटेड, मतलब ये खत्म होने वाली है थोड़ी टाइम के लिए. अब इसके 5 phases थे that we studied. These are the 5 phases. First is lactoname. यहाँ पर प्रोफेज जो है बिगिन हुआ था. क्रोमोसोम स्टार्ट टू कंडेंस. यहाँ पर हमें थोड़े से क्रोमोसोम दिखने शुरू हुए थे. अर्लियर इट वाज लाइक ए स्ट्रैंड, जैसे धागे होते हैं ऐसे ऐसे नजर आ रहे थे. ओके, आफ्टर लैक्टोनेम जाइगोटेम. एंड इन जाइगोटेम व्हाट हैपन? अगर ये क्रोमोसोम एक है, ये क्रोमोसोम दूसरा है, सो दे स्टार्ट्स टू क्या हुआ? आपस में मिलना शुरू हुए थे साइडवेज. एंड व्हाट दे मेड? इस कॉम्प्लेक्स को क्या कहते हैं? क्या कहते हैं बेटा? सुनाई दे रहा है मुझे, बट हु इज स्पीकिंग, हु इज टेलिंग? बेटा जिसको आता है जल्दी बताओ. ऐसे विस्पर क्यों करते हो? व्हाई डू विस्पर? बताओ बेटा. क्या कहते हैं इसे? अच्छा मैं प्रोसेस का नाम बताती हूँ. आई एम गोइंग टू टेल यू द नेम ऑफ द प्रोसेस. साइनेप्सिस. इट इज साइनेप्सिस. So do you remember the complex name now? Yes. What is the name of the complex? Synaptominal complex. ठीक है. So here synapses begin. यहाँ पर देखो sideways you can see they are aligning. ठीक है. They are making synaptonemal complex here. After that you can see now strands are exchanging. They are exchanging here. ठीक है. यहाँ पर exchange का जो event है it gets started over here. Means crossing over started over here. Crossing over means recombination. Exchange of genes. Is fragmented for genes will go to this fragment, okay, of another chromosome. ठीक है. So here exchange of genes in chromosomes. ठीक है. So recombination is happening where? In this. Where it is happening? In this. That is pachytene. In diplotin only. Where now synaptominal complex will dissolve. जो side base जो उनकी joining थी वो खत्म हो जाएगी. They will only join at one end. जहां पर recombination event हो रहा था. That is chiasmata. सिर्फ that end. And that basically called what? Diplotin stage. वो diplotin stage बताता है. Dikinesis में now they both are separate from each other after mixing of their genes. Okay. So now tell me कहाँ पर recombination जो है the event or we can say recombination event is completed. कहाँ पर complete हुआ? Where does it complete? Is it pachytene or diplotin? ये देखो it is in front of you. Just go through this image. So where recombination is completed? Yes, beta. Where does this recombination completed, beta? पिन ड्रॉप साइलेंस. हाँ. अब तो मैंने बोला भी नहीं कि पिन ड्रॉप साइलेंट हो जाओ. क्वेश्चन पूछ लिया मैंने. Yes. Where it is completed? Okay. Middle row, second bench girl. Yes. Corner girl. Yes, बेटा खड़े हो जाओ. Yes. Pachytene. Why pachytene? Okay. So reason is this beta because crossing over recombination is happening here. In this, it's not happening. It's ending. Ending means only the point where they already did their recombination. Only that point they are bind. Otherwise, they are separated from each other. You can see the recombination is already happened. You can see there is exchange of genes. Now this red color gene is here on this chromosome blue and blue is on the red. It's already happened in the previous, that is pachytene. So answer is D. Write down beta, answer is D. Coming to C, seventh question, the dissolution of synaptonemal complex occurs during. कौन से कॉम्प्लेक्स कौन से इसमें जो है अब सिनेप्सिस आपको देखने को नहीं मिलेगी. दे आर सेपरेटेड. कहाँ डिसोल्यूशन हो रहा है, कहाँ पर वो अलग हो रहे हैं. विच में? Diplotin. In diplotin, we can see that dissolution of this synaptonickel complex happens. So it's seventh का है B. Write down answer is B. Okay. During which stages of mitosis and meiosis, respectively, does the centromere of each chromosome split? Centromere. अगर centromere split हो रहा है, it means they are making sister chromatids. So sister chromatid, one is going to one end of the pole and second is going to the another end of the pole. जो भी हमने डायग्राम बनाया था. ध्यान से देखो. There are two stages of meiosis, meiosis 1 and meiosis 2. ठीक है? In meiosis 1, there is no sister chromatids that form. Meiosis 1 में sister chromatids अलग नहीं होती क्योंकि उसमें chromosomes हमारे पास 2 हैं, chromosomes अलग होते हैं. ठीक है? In meiosis 2, as we learned PM at, ठीक है? It is the only anaphase, whether it's mitosis, whether it's meiosis. In meiosis 2, I'm talking about. It's only the anaphase where you can see that sister chromatids are moving like this. Okay? It's only in the anaphase. Now, which anaphase of the meiosis? It's 2. It's 2. Okay? And in mitosis, we don't have 2 sets. We have only 1 set of anaphase. So it's we can say simply anaphase. Okay? So in meiosis, we say anaphase 2, and in mitosis, it's only anaphase. So what could be the answer here then? Yes, beta? D. So answer is D. Okay? Its answer is D. So can you tell me what happens in meiosis 1 anaphase then? It is chromosome. Okay? So now, next move to the question number 9. Which of the following stages of meiosis involve division of centromere? So I hope you can get the answer of this. Anaphase 2. So answer is, that's really good, beta. That's really good, B. Coming to 10th question. The process of appearance of recombination nodules occurs at which substage of prophase 1 in meiosis? Pachetin. Yes, it's pachetin. A is the correct answer. Coming to 11th, which stage of meiosis lasts for months or years in oocytes of some vertebrates? Yes, beta? So it's D. Okay? No doubt. Coming to 12, in meiosis, crossing over, exchange of material between homologous chromosomes is catalyzed by the enzyme. Which beta? Recombinase. It's C. Yes, it's C. Coming to 13, the appearance of recombination nodules on nodules on homologous chromosome during meiosis characterizes what it shows if recombination nodule forms. Bivalent. Bivalent तो पहले बन गया. That is synapses वाले part में. That structure is called bivalent. Sites at which crossing over occurs. recombination नोड्यूल बन गया, इसका मतलब यहां पर ही अब क्रॉसिंग ओवर होगा. ठीक है? Coming to 14th question, which stage of meiotic prophase shows terminalization of chiasmata as its distinctive feature? Trikinesis. C. That's good. 15. Bivalent or tetrad formation is characteristic feature observed during. अभी मैंने जस्ट थोड़ी देर पहले ही बताया था. आई थिंक 2 और 3 को बताया. बाइवेलेंट कब बनता है? इन विच फेज? बी बेटा. सी नहीं, बी. बी बाइवेलेंट. बाइवेलेंट फॉर्म से zygote where synapses take place. ठीक है? So it's synaptonemal complex in zygote stage. It's B. It's B. ठीक है? Now coming to the 16, match the following columns with respect to meiosis. So first is saying, so it's a basically events. Okay. So column 1 is zygote. So what happens in zygote out of these? Yeah. What happens in zygote? Synapses. ठीक है? Synapses. That's nice. Now coming to pachetin. बेटा Pachytail crossing over. Yes, crossing over. Now diplotail. Yes, Tasmata. And the last is we left with odd one out, that is dikinesis. So obviously is communalization. ठीक है? So according to this option, which option is correct? A. That's right. So 17, 18, 19 we are not going to do because these are repeated questions. ऐसे ही कुछ क्वेश्चंस पीछे भी हमने कर लिए और आगे करेंगे. So we are not attempting 17, 18. Write down उनके आगे लिख दो repeated, repeated, repeated. 17, 18, 19. Almost repeated. ऐसा नहीं कि सेम ही लैंग्वेज होगी. Wordings can be different, but one of the same thing. ठीक है? So I want you to attempt these questions itself. Now coming directly to the 20th question. Given below is the representation of a certain event at particular stage of a type of cell division. Which is this stage? कौन सी स्टेज है बेटा? कौन सी ऐसी स्टेज है जिसमें जो है we can observe this event? कहां पर हमें ये चीज देखने को मिलती है? In which stage? Prophase 1 of during meiosis. So it's A. ठीक है? It's A. Now 21st question. Two statements are given. You have to tell is it right or wrong. ठीक है? Statement 1. Chromosome become gradually visible under light microscope during lactotein stage. Yes. ठीक है? That's correct. Statement 2. The beginning of diplotin stage is recognized by dissolution of synaptonemal complex. हाँ सही है, ये भी डिप्लोटीन में होता है ना. So that's correct. Both are correct. 22nd question. Select correct sequence of substages of prophase 1 of meiotic. So अभी हम ये वाला भी नहीं करेंगे. This is repeated. 22nd is repeated. 24th is also repeated. 24th is also repeated. ठीक है? These are the questions that you are going to attempt itself. ठीक है? Coming to 23rd question में आ जाओ. Select the correct statements. First statement is क्या बेटा? Tetrad formation is seen during lactotein. Is it correct? Where it can be seen? Zygote. That's right. It can be seen in zygote. During anaphase, the centromeres split and chromatids separate. Yes, is it correct or not? कैसा होता है? Yes. So in anaphase only you can see the splitting of centromeres and you can see the here the sister chromatids. So that's correct. Okay. So it's an anaphase. So it's correct. Second statement is correct. Coming to C, terminalization take place during patchety. It takes place in which phase? Dikinesis. It takes place in dikinesis. Now D, nucleolus Golgi complex ER reformed during telophase. Is it correct? दोबारा से न्यूक्लियस बनता है, एंडोप्लाज्मिक रेटिकुलम आता है, गोल्जी आता है. क्या वो टेलोफेज में आता है? यस. एनाफेज के बाद टेलोफेज आता है, उसी में आता है. वी कैन से टू डिफरेंट न्यूक्लियस इन दैट. ठीक है? सो इट्स ट्रू. This statement is true. Going to E, crossing over take place between sister chromatids of homologous chromosome. Yes. किसने पहले हाँ किया? पहले तो ये बताओ. हाँ, सिस्टर क्रोमेटिड्स नहीं पता? ये देखो, ये सिस्टर, इन दोनों के बीच में थोड़े ना हो रहा, इन दोनों के बीच में हो रहा है, एंड दीज आर नॉट सिस्टर्स, कजन्स है, सिस्टर्स नहीं है, ठीक है? सो नाउ, so answer according to this is B and D are only correct, so option A is correct, option A is correct, ठीक है? So coming to 25th, I think 25th you can do yourself. This is again repeated. वही की event, chromosome कहाँ दिख रहा है, crossing over कहाँ हो रहा है. So 25th you can do yourself. Do the 25th also. ठीक है? Now coming to 26. If a cell has chromosome number 20, number of chromosomes in G2 phases are. क्या क्रोमोसोम का नंबर चेंज होता है? Does chromosome number change in cell division or cell cycle? Yes or no? तो बेटा इट्स फॉर जस्ट फॉर कंफ्यूजिंग यू, ये आपके पेपर में भी क्वेश्चन आ सकता है, ठीक है? सो क्रोमोसोम नंबर नेवर चेंज इन सेल साइकिल. इफ इट स्टार्ट फ्रॉम द 2n इट रिमेन 2n, ठीक है? तो मियोसिस को छोड़ के इट इज 2n, ठीक है? इट इज 2n. अगर यहाँ पर ये 20 है तो आफ्टर G2 में भी इंटरफेस की बात कर रही हूँ, आई एम टॉकिंग अबाउट इंटरफेस. ये 20 ही रहेगा, इट विल रिमेन 20 इन G1 S G2, इट विल रिमेन 20, ठीक है? सेल डिवीजन से पहले की बात. नाउ कमिंग टू 27. इफ अ सेल इज इन I phase मींस इंटरफेस फेज में है, द DNA अमाउंट इन G1 एंड G2 फेज इज, क्या होगा? व्हाट विल हैपन? ये हमारे पास सेल साइकिल है, दिस इज सेल साइकिल, ये हमें पता है, दिस इज M फेज, दिस इज G1 फेज, दिस इज S फेज एंड दिस इज G2 फेज. तो नाउ G1 फेज और G2 फेज का DNA का अमाउंट सेम रहेगा. यस. if I say just take an example beta, if I say यहाँ पर हमारे पास 10 gram DNA, just I'm taking an example, it's not in gram, so just take an example. If we have 10 gram of DNA here in the G1 phase, so G2 में क्या होगा? Is it 10 gram or less or more? Yes बेटा, ऐसे नहीं, whispering no whispering, just tell me whatever you know, just one student come and speak up. क्या होगा? What will happen? I can see students wants to participate but not individually, in a group. Why so? Why so? Okay, middle row, fourth bench, second girl. खड़ी हो जाओ बेटा. Fourth bench, yes, second girl. खड़े हो जाओ बेटा, stand up. Now tell me, if we have DNA of 10 gram at G1 phase, ठीक है? तो G2 phase में कितना DNA होगा? Is it same, less or more? Yes बेटा? बेटा tell me, speak up. Whatever you know, just tell me. Will it remain same, increase or decrease? I'm not able to hear you. Come forward. Come forward. And rest of the students, please be quiet. Let me hear her. मुझे सुनने दो. हाँ यस बेटा, यहीं खड़े हो जाओ. Increase. कितना increase होगा? 10 ग्राम और increase होगा तो कितना बन जाएगा? 20 ग्राम. 20 ग्राम. That's nice. That's nice. Why it becomes 20 ग्राम here? क्योंकि ये तो फेज बीच में आया, एस फेज, जहां पर डीएनए की रेप्लिकेशन हुई. डीएनए रेप्लिकेशन मींस एम्प्लीफिकेशन ऑफ डीएनए. डीएनए की क्वांटिटी इंक्रीज हुई, डबल हुई. सो दैट्स व्हाई जो G2 फेज जितना डीएनए था उसका डबल हो जाएगा इन G2 फेज. इट्स ओनली एप्लीकेबल टू द डीएनए, नॉट फॉर द क्रोमोसोम. यस बेटा, दैट्स गुड. सिट डाउन. नाउ द आंसर ऑफ दिस इज बी. 27 का बी. G1 same as parent cell, G2 double as compared to parent cell. Okay. Now coming to 28. Now it is from your NEET exams. प्रीवियस ईयर के क्वेश्चन पेपर में से है बेटा, पे अटेंशन। अब ये जो क्वेश्चन है ये उनमें से है कुछ जो मैंने आपको मार्क किए हैं रिपीटेड, ठीक है। Regarding meiosis, which statement is incorrect? Regarding meiosis, which statement is incorrect? Now, but what shall I say? Sorry, is more excited to tell answer. Answer. So how can sir be? Now the question is how can sir be wrong? Nahi na? So answer is correct. It's A. So DNA replication occurs in S phase of meiosis 2. So it's A. Okay. There is no DNA replication in meiosis. That is in S phase of cell cycle during interphase. ठीक है? Coming to 29th question, NEET exam again. Select the correct sequence of events during prophase 1. Means we are talking about 5 phases. ठीक है? तो जो synapses है, कौन कौन सा order? You have to tell the order. To tell the order, what you need to do, बेटा? Firstly, फटाफट write down. ठीक है, फेजेस नेम, अगर फेजेस के नेम पता होंगे यू कैन इजिली रिकॉल द इवेंट। तो क्या है? साइटोसिन, दिस इज द शॉर्ट, बेटा दिस इज द शॉर्ट। ठीक है, टिप्स मैं आपको बता रही हूँ हाउ टू सॉल्व क्वेश्चन इन नीट एग्जाम। नीट के हिसाब से बता रही हूँ, तो फोकस, प्लीज पे फोकस, ठीक है, अटेंशन। ये आपके नीट में काम आएगा कि अगर कभी भी कोई इवेंट से रिलेटेड ऐसा क्वेश्चन आता है जिसमें मल्टीपल इवेंट्स आर इन्वॉल्व, डू राइट लाइक दिस। ठीक है, फर्स्टली राइट डाउन द नेम बिकॉज वी गेट कंफ्यूज, बिकॉज उस टाइम हमारे ऊपर थोड़ा प्रेशर होता है टू सॉल्व द क्वेश्चन। ठीक है, सो राइट लाइक दिस, फटाफट अपना लिखा। देन फटाफट इसमें क्या होता है, इसमें आप ऐसे लिखो क्रोमोसोम, सिंपल ऐसे, जाइगोटिन सिनेप्टो, ठीक है, शॉर्टकट। पैचेटिन क्या हो रहा है हमारा, जो है रिकॉम्बिनेशन, खत्म। डिस्कोटिन क्या हो रहा है, चेस्मेटा, खत्म। ठीक है, डाइकाइनेसिस क्या हो रहा है, टर्मिनलाइजेशन। नाउ यू हैव एवरीथिंग इन फ्रंट ऑफ यू। आपको अपनी राइटिंग खुद समझ आ जाएगी ऐसे शॉर्ट टिप में जब लिखोगे। ओके, तो जब यू व्हेन यू राइट दिस, नाउ रीड दिस क्वेश्चन। सो साइनेप्सिस, वेयर साइनेप्सिस एट अकरिंग, कहां पर हो रहा है? So this is the A. Okay. Now coming to chromosome visibles. Where chromosomes are visible? Yes. Where chromosomes are visible, beta? I think this is given in sequence only. No. Just give me a minute. Okay. Yes. So chromosomes visible. सबसे पहले क्या होगा, हमारे क्रोमोसोम विजिबल्स कहाँ होते हैं? It's in this. ठीक है, क्रोमोसोम विजिबल्स हो गए। क्रॉसिंग ओवर, वेयर क्रॉसिंग ओवर टेक प्लेस? ठीक है, देन terminalization of transmitter, where it takes place? And we left with E. So now ये कहां पर आ रहा है ऐसा? तो अब जब ऐसा करोगे, देयर आर वेरी लेस चांसेस ऑफ अटेम्प्टिंग क्वेश्चन रॉन्ग। चांसेस तो बहुत ही नेगेटिव चले जाएंगे। ठीक है, और जब आप एक बार ये बना लोगे, ऐसा और भी क्वेश्चन होगा तो यू कैन इजीली सॉल्व इट। ठीक है, कमिंग टू द असर्शन रीजन। Now assertion is saying meiosis causes variations in the daughter cells form. क्या वेरिएशन होती है? Do we see any variation in the cells? क्या वो एक जैसे होते हैं, एकदम रेप्लिका बन के आते हैं? अगर मेरी ये बॉटल है, वो वैसे ही एक दो और बॉटल्स बन जाएंगी, वहां पर उसमें कोई डिफरेंस नहीं होगा। ऐसा टेल मी, इज देयर एनी डिफरेंस? बेटा इसमें रिकॉम्बिनेशन हो रहा है। वी हैव सीन रिकॉम्बिनेशन, एक्सचेंज ऑफ जीन्स। ये देखो, फिर नेक्स्ट हमने क्या देखा था, व्हाट वी नोटिस्ड हियर, वेरिएशन थी या नहीं थी? अगर वो एक जैसा रहेगा ही नहीं, उसमें जो है कुछ हम चेंज कर देंगे, सो देयर ऑब्जर्व वेरिएशन एंड इट हैपेंस इन मियोसिस. ठीक है. सो दिस स्टेटमेंट इज करेक्ट, assertion इज राइट. रीजन, crossing over exchanges segments of DNA between non-sister chromatids. Is it right?
Yes.
Yes, it's correct. It's correct. ठीक है. Coming to 31st question, assertion. Meiosis 1 is called as reduction division.
Yes.
Reason, DNA content is reduced to half as compared to parent cell after meiosis 1. क्रोमोसोम नंबर घटता है या डीएनए कंटेंट घटता है? क्योंकि जब हम मियोसिस की बात करें, वी टॉक अबाउट मियोसिस, यू नो अबाउट, वी स्टार्ट फ्रॉम द डिप्लॉइड सेल एंड व्हाट we get 4 daughter cells and those are haploid. That is what we get, है ना? So what happens in that? DNA content get decrease or chromosome number get decrease? What happens?
Number gets decreased.
Are you sure? क्योंकि मैंने कंफ्यूज कर दिया सबको, एक और ऑप्शन दे दिया. नाउ स्टूडेंट्स आर कंफ्यूज एंड दैट इज व्हाट नीट के जो पेपर के एग्जामिनर्स हैं, जो बनाते हैं पेपर सेटर्स, दे डू. भी ऐसे ही देंगे. दे आल्सो गिव यू लाइक दिस choices. So yes, chromosome number is getting decreased. If you notice, from 2n it becomes n, diploid to haploid. Number decrease हुआ ना? Otherwise we have 2 sets of chromosome. At the end we have only 1 chromosome, and that makes our daughter cells haploid. ठीक है? Is it correct? Correct? Understood? Okay. So I think time is over, beta. So try to solve 32 to 35 itself because these are the ones we already attempted. ठीक है? ये कर लिया. So any doubt, beta? किसी को कोई doubt? Any doubt? That's good. That's good. You such a wonderful students. हाँ? Fully participate. अच्छा लगा. Participate करा करो ऐसे ही जैसे आज किया. ठीक है? So that's good. Thank you, sir.